In this section, we're going to take a look at signal transmission at the synapse. You'll remember when we discussed the muscular system that we dealt with one particular kind of synapse, the neuromuscular junction, the location where a motor neuron will communicate with a muscle cell. In this chapter, we're going to focus on the type of synapse that involves neurons communicating with other neurons. There are two types of synapses, electrical and chemical. In this chapter, we'll focus on the chemical synapse. But first, let's mention just a little bit about what are called electrical synapses. These are the types of synapses that involve an ionic current spreading from one cell to the next cell through something called a gap junction. A gap junction is simply a location where one cell is kind of attached to another cell, and there's a hole that's found between the two cells, and the ions can flow directly from one cell to the next. These kinds of synapses allow faster two-way transmission, and this also allows for there to be a synchronization of groups of neurons. These kinds of synapses are also found in visceral smooth muscle, cardiac muscle, and in developing embryos. So if, for instance, you're talking about cardiac muscle, what this allows to happen is for an electrical message to travel very quickly from one cardiac muscle cell to the next cardiac muscle cell so that the muscle cells will all contract in synchrony. Most of this section we'll deal with what are called chemical synapses. With this kind of synapse, there's one-way information transfer from what is called a presynaptic neuron to a postsynaptic neuron. There's three kinds of synapses that are chemical synapses: an axodendritic, in which the message is traveling from the axon of the presynaptic neuron to a dendrite of the postsynaptic neuron. Another kind of synapse is one in which the message is going from an axon to the cell body. And although we usually don't talk too much about these, there are other kinds of synapses, ones in which an axon communicates with another axon. In this series of pictures, which are very important, we're going to look at the series of events that take place at a chemical synapse. As you can see from this diagram, we have a variety of numbers that indicate the sequence of events, and in each of the following slides we will discuss what happens at these various steps. With step 1, a nerve impulse, that is an action potential, is traveling down the axon, and it has arrived at the synaptic end bulb of the presynaptic axon. When that action potential reaches the synaptic end bulb, then the depolarizing phase of that action potential will cause voltage-gated calcium ion channels to open up. These calcium ion channels, as you can see in this diagram, are embedded in the membrane of the synaptic end bulbs. And because there's a greater concentration of calcium ions outside of the cell, the opening of these channels will allow calcium ions to move in. When these calcium ions move in, that increase in the concentration of the calcium ions will trigger exocytosis of the synaptic vesicles. The synaptic vesicles, you'll remember, are these little pockets that contain neurotransmitters. And so when the synaptic vesicles migrate toward the edge of the plasma membrane and perform exocytosis, the neurotransmitter molecules that are in the synaptic vesicles are released into the space called the synaptic cleft. The neurotransmitter molecules then bind to neurotransmitter receptors, which are found in the postsynaptic neuron's plasma membrane. In this particular picture, the receptor is a ligand-gated channel, and what that means is that the neurotransmitter can attach to it. There are other kinds of receptors, but we'll focus on these kinds of receptors right now. When the neurotransmitter molecules bind to the ligand-gated channels, it will cause those channels to open up. And when they open up, then ions will flow through the membrane. In this particular picture, we can see sodium ions flowing through the ligand-gated channel. But they can be different kinds of ions depending upon the type of neurotransmitter and the type of receptor that we're talking about. As the ions flow through the membrane of the postsynaptic neuron, it will cause the membrane potential of that postsynaptic neuron to change. That change in the membrane potential of the postsynaptic neuron is called a postsynaptic potential. Now, that postsynaptic potential could either cause hyperpolarization or depolarization. If the particular receptors or ligand-gated channels are sodium channels, then sodium ions will rush in. And since sodium ions have a positive charge, that will make the inside of the cell more positive. So in other words, this will cause depolarization of that postsynaptic neuron. If, on the other hand, the ligand-gated channel is one that is for either a chloride or a potassium channel, then what will happen is the postsynaptic neuron will undergo hyperpolarization. And that is because there's a higher concentration of chloride ions that are outside of the cell. And so when the chloride ion channels open, the negative chloride ion will move inside of the cell, causing the inside to become more negative, or in other words, hyperpolarize. If, on the other hand, the ligand-gated channel is a potassium channel, then this will cause the potassium ions to diffuse out. You'll remember that there's a higher concentration of potassium ions inside of a cell. And so whenever we open up these channels, the potassium ions will diffuse out. Since potassium ions have a positive charge, then when they leave the inside of the cell, that will make the inside of the cell less positive. With the inside of the cell becoming less positive, that's another way of saying it's more negative. And in other words, this will cause hyperpolarization. If a depolarizing postsynaptic potential is at the threshold level when it reaches the trigger zone, which is near the axon hillock, then an action potential will be generated in the axon of the postsynaptic neuron. So in other words, what we're saying here is, let's suppose that depolarization occurs at this site of the synapse. That depolarizing postsynaptic potential will then travel down the surface of the dendrite or the cell body. And remember, this is going to be a graded potential. And so it will decrease in its amplitude as it moves further and further away from this source of stimulation of these neurotransmitters. When that graded potential reaches the axon hillock, or specifically the trigger zone that's near the axon hillock, if the inside of the cell is at the threshold level, approximately -55 millivolts, then that will generate an action potential at the beginning of the axon.